thank you all for attending this seminar. I'm introducing Dr. Nico Franzmeier from LMU Munich University Hospital. Nico, I learned today that he did his studies in medicine in Austria. I didn't know that. But after that, I think all his career he has been in Munich, where he reached the PI position very young, I think 33, 34, right, Nico?
31 back then, yeah.
So one of the people in the field that got to a PI position quicker, I think that I know. He has also been awarded with several awards, including the De Leon Award from the Alzheimer's Association for his research, and he will be presenting his main research line today that I think has, you will see some very cool examples on how we can use imaging, not only for diagnosis or disease tracking, also to understand how the disease mechanisms work. And I hope you find it very interesting. And that's all for me. Nico, the stage is open.
Thank you, Jesus. I think you need to turn that off, otherwise we may have feedback. No? Yeah, is it? Okay, good. Yeah, we've just had lunch. I hope I'm not too tired, and I got up at 4:15 a.m. this morning in Munich. So yeah, Michel, who can't unfortunately be here, was one of the first people I got to know in AD research, basically, who familiarized me with many people and helped me form my network. So I'm very happy to be here. Unfortunately, he couldn't be here. So I'll try to give an overview of the work that we've been doing, I would say, over the last 6, 7 years or so, on still some of it from my postdoc time. On mechanisms of tau spreading and in Alzheimer's disease, and with a short outlook to primary tauopathies in the very back. So you have to obviously start any talk with Cliff Jack's biomarker curves. It's just to emphasize again how we currently understand the cascade of pathological brain changes, and I'm trying to pinpoint to the different research aims that we're trying to follow in our group. So basically, the whole thing kicks off with the brain-wide accumulation of beta amyloid pathology, which is then followed by a gap of a few years that's inter-individually quite heterogeneous, by the onset of tau pathology, typically somewhere in the temporal lobe from where it travels. And then once you have tau, you develop typically neurodegeneration and then a spectrum of cognitive decline, all up to the level of dementia. And I'm trying to cover a few things today. First of all, how does tau pathology spread? And which factors can we identify which may accelerate or attenuate its spread? And then, most important of all, why does that happen only in the presence of amyloid, at least mostly for AD type tau pathology? And to that end, we're using PET imaging mostly because we need the spatial information of where the tau pathology or the pathologies are in general, which we combine quite heavily with connectomics. You'll see in a minute why. And obviously, you can't get away today without using good biomarkers. So first things first, how does tau spread? And I hope that ringing stops in a minute. So what do we know about tau? Tau is a physiologically highly abundant protein in all of our brains, within neurons typically, where it helps stabilize microtubules. So the axonal pathways, the highways basically of the cell, which can be fairly long. What happens in Alzheimer's disease is they become hyperphosphorylated, form first of all seeds, then protofibrils, finally tangles, and so on. Microtubules disintegrate and the cell dies. So in very simplified terms. And one of the very important pathways that has been identified for tau to basically aggregate or spread throughout the brain is via taking the synapses as a key route, because it's an intraneuron protein. So it has been shown that tau can go into synaptic vesicles and basically by that enter downstream cells via different pathways that are shown here. So why is tau so important? You can have loads of amyloid in your brain, but don't have any signs of dementia. That's what we consider the stage of clinical AD. But once you have tau, you're very likely to show clinical decline or any sort of cognitive impairment. So there are very few cases with a brain full of tau pathology who have no symptoms at all, meaning tau is really predictive of clinical progression and of your clinical phenotype. So this is why we, back then when I got my hands first on tau PET imaging data, tried to understand why and how does that spread. And basically we formed this connectivity-based tau spreading model. So you lay out the brain's network. It's kind of the roadmap of the brain, like literally roads cover or connecting different cities in Spain, let's say. And we know that tau typically starts very focally somewhere in the brain. And then we thought, okay, if it spreads across connected regions, then the layout of the brain network, so the number of connections between any given brain regions, should tell us about the spatial pattern of how tau propagates and spreads. This is why we make heavy use of connectomics to map out the basic brain network topology, which we then combine with tau PET imaging. Most of it is for tau superior data. And longitudinally, ideally, to really model the spread over time. And this is from two of our first publications using tau PET, which is now, 7 years ago, the first one at least, where we got longitudinal tau PET data. Back then, it was just 40, 50 individuals that were in ADNI. Today, it's 300, 400, which we combined with fMRI-based connectivity. And in order to simplify things a bit, we typically parcel the brain into a set of brain regions, covering only the neocortex to exclude all the regions where there's off-target binding. And then we first used fMRI to compute the functional connectivity between any two region pairs here. So this is just everything in darker blue here means there's a stronger connection between those brain regions. And then we did a very similar thing for tau PET, which is looking at the covariance in tau accumulation over time. So here we looked at so let's say you have the left angular gyrus and the right angular gyrus. You compute a change rate of tau for both regions, and then look at how correlated are they. If they accumulate tau together, so to say, then they should get a darker color in that matrix. And then if you put those in a scatter plot, you see that brain regions who show a stronger connection to each other, show more similar rate of tau accumulation. That is kind of take home number one. Like if you consider the Hebbian rule, what fires together wires together, we here say what fires together accumulates tau together. And because reviewer 2 wanted it, we had to contact Oscar in Sweden and replicate everything in Biofinder 1, which luckily we managed to do. And that was kind of our first take on, okay, the brain network architecture does have something to do with the pattern or the rate in which tau accumulates. So can we use connectivity to forecast how tau spreads? We recently then extended this to a more heterogeneous data set together with Rick Ossenkoppeler from Amsterdam and our joint PhD student Hannah, and tried to collect all atypical AD cases that we could get from our international collaborators. So it's so far, I think, the largest atypical AD database that exists. And this is just where their tau pathology typically starts. So in posterior cortical atrophy, a visual phenotype, you have it more in the back of the brain, a language variant more on the left side, so language dominant hemisphere, and so on. And we repeated the entire process. And basically, now here the scatter plots are inverted because we transformed connectivity to distance. Everything that is more on the left has a closer connection, so to say. You see the same thing. For all the different phenotypes, the closer two regions are connected, the more similar their tau accumulation. And this was just to generalize across spatially heterogeneous patterns, because in the previous study, we had typical amnestic patients which share this common temporal lobe pattern of tau pathology. So then we moved to the next step and thought, can we predict on the individual level how tau propagates and spreads? So can we take a patient's image at baseline, define where does he or she have highest tau pathology at that given point in time? So denote their epicenter, and then group the remaining brain into 4 big chunks, Q1 for quartile 1 to Q4, based on how well they are connected to that epicenter. It has nothing to do with spatial proximity, only connectivity-based proximity. And our rationale was that in those regions that are really closely connected to that epicenter, we should see fast tau accumulation. And the further you go away from that tau epicenter, the less tau accumulation you should observe. This is in essence how these regions look like. And they are, that is important, determined for each patient individually. So we're not taking group level Bragg masks, which do not capture that inter-individual heterogeneity. And you can see this is just a probabilistic mapping that the epicenters typically cluster around, somewhere around the temporal lobe, but everyone who's looked at a substantial amount of tau PET images knows, and you've done visual reads, uh, it's not all Bragg. So people deviate from it quite substantially, but you see downstream, then this Q1 region clusters around what looks a bit like the default mode network, and then if you go all the way to the end, you basically have what we would consider Bragg 6 region. So, but you capture that individual heterogeneity. So this is where we would expect most tau accumulation to happen. And in fact, back then in that study, it was still a limited sample. Now we have 5 times the amount. But always in this Q1 region, we see for each individual the highest tau accumulation, regardless of the sample we've been looking into. And every subject is represented in each of those box plots. So you can really determine on the individual level, zoom into the brain by knowing where it starts, knowing the connectional architecture of the brain, we know or we can predict where it goes. And same thing again here for our atypical cases. This is cross-sectional data only, but here it works as well. So we can, regardless of phenotype, we can predict where tau should be highest, regardless or from next to the, if you will. Why is that important? Is that only an academic exercise? I think no, because let's say you have a tau targeting intervention. Let's say these silencing approaches where you silence the messenger RNA. Then you want to see, is there an attenuation of tau spread? Then you need to know where to look at into your brain. And with our region that we tailor to each individual, we think this is a simulated clinical trial. You can get a higher sensitivity in order to detect here potentially a simulated treatment effect compared to if you use Bragg staging ROIs or any meta ROI that does not consider inter-individual heterogeneity, which is really substantial for tau. So that is lesson number 1. How does tau spread throughout the brain? Next thing is, why does that happen really only or mostly in the presence of amyloid? Because we know from a lot of post-mortem studies, which you conduct here in-house quite a lot, as I've learned today, that as we age, most of us develop some sort of tau pathology in their brainstem or in their temporal lobe somewhere, hippocampus or entorhinal cortex. But if you don't have amyloid in your brain, it typically stays there and doesn't go much further beyond, which is what we histopathologically term PART, primary age-related tauopathy. And at the amyloid imaging conference now in Puerto Rico, which happened just 2 weeks ago, there was a debate of whether a Bragg 5 case can even be considered PART, because that's by definition, it's a stupid discussion, but you see where this goes, that you can have, or that tau pathology in the absence of amyloid is typically fairly restricted, but if you have amyloid in your neocortex, this tau pathology is really amplified and spread. So amyloid is like putting fuel in the fire and leading the fire to burn quicker throughout your brain. So how does that happen? How does amyloid do that? Here we can look at biomarkers again, and biomarkers not as just diagnostic tools, but as tools that their name suggests, indicators of biology or abnormal biology. And if you look here as amyloid as a cause of the whole thing, here on the x-axis, that's just amyloid PET, and tau pathophysiology markers on the y-axis. This is p-tau217, so now a T1 biomarker, and tau PET, a T2 marker, fibrillar tau pathology. And you see that p-tau goes up much earlier. You see fibrils much later. That may have to do with sensitivity of the tools that we use, but it may also have something to do with biology, that this p-tau response is a gain of function, that amyloid neurons start secreting phosphorylated tau when amyloid is present. That has been shown by the group of Randy Bateman, that this p-tau is not just a spill out from dead neurons, it's actively synthesized from living cells. So they secrete it. And this is what we pick up with our extracellular pool, either in CSF or in plasma. So our idea of the whole cascade is that you have amyloid accumulation triggering hyperphosphorylation of tau or just coinciding with that, but that amyloid leads to a synaptic release of p-tau. Why and how, I'll show that later again. Amyloid is a strong driver of neuronal hyperactivity. This is why so many people in early AD stages show, if you look closely, subclinical epileptiform activities. Some even manifest seizures. Lots of work by Mark Busche has shown that. And if phospho-tau goes into a synaptic vesicle, where the fusion with the presynaptic membrane is obviously activity dependent, then this might be a pathway linking the two. So amyloid triggers some slight neuronal hyperactivity, therefore more p-tau is released, and we see its increase in CSF. And that this p-tau is potentially the pathological force leading, which is secreted to the infection, if you will, of the downstream neuron, and then to a cascade of spread. So we wondered, can we actually test that in vivo? And we thought, okay, this pool of secreted p-tau is accessible in biofluids. Back then here we used CSF. So is the association between having fibrillar amyloid and fibrillar tau pathology mediated or driven by that intermediate step of phospho-tau, which is now kind of in the framework as T1 and T2 biomarkers? So this was work done together with Alexa Piché-Binet from Biofinder, and we've looked into how does amyloid predict where your tau accumulates. So this is all Biofinder 2 data. And amyloid does a fairly okay-ish prediction. Everything in red, there we find a significant prediction of you have amyloid pathology at baseline, you'll accumulate tau longitudinally in the future. If you look at p-tau instead, the entire brain is red. So p-tau is a much better predictor of how fast your tau accumulates than amyloid alone. So if you put both of them in the exact same regression equation, the effect of amyloid completely disappears. Only p-tau is left explaining variability. Now that does, of course, not mean amyloid is irrelevant, but it means, and you can formally test that, what we did here, a mediation effect, like we anticipated, that the effect of amyloid on tau accumulation is mediated by that intermediate increase in p-tau. And this is for us kind of a key take home, that breaking that part here from amyloid to p-tau secretion could also be a potentially good way of interfering with tau accumulation, as long as tau accumulation is amyloid dependent. So it becomes amyloid independent at some point, which I don't have slides here, but we're working on that. So that's one part of the amyloid story. I'll come back to that later on. But now that we've established that more or less quasi-mechanistic framework of how we can study tau accumulation and spread across brain connections, we can look at which factors modulate its spread, so which accelerate or decelerate it, in order to get a better idea of where could be potential therapeutic angles that could help break that amyloid to tauopathy transition. So an obvious one is looking at genetics, which back then PhD student in my group did. And she was very interested, she's now a postdoc, looking at ApoE4, which all of you know is the main or the strongest risk factor for sporadic AD and the most prevalent one. And if you think of that transition from having amyloid to having tau to developing neurodegeneration and a manifest clinical syndrome, ApoE4 has long been known to promote amyloid pathology. So these people develop amyloid to a stronger degree and at a younger age. But is there also an effect on the transition to tauopathy? It was kind of an exploratory approach here. So we looked again, you only basically need to look at the leftmost plot. This is this individualized readout, so this Q1 region where people should accumulate tau fast. And that plot puts amyloid pathology at baseline against the rate of tau accumulation. And everyone in blue is an E4 non-carrier. And you see, okay, they start developing or accumulating tau fairly late, when there's already substantial amyloid pathology in the brain, and then shoot off. And in orange, the E4 carriers shoot off much earlier. In fact, their increase is significantly different from 0 in longitudinal tau PET, when we would, from a quantitative point of view, consider them still amyloid PET negative. So you see here a dissociation, that the effect of amyloid on tau is stronger in E4 carriers. And we've recently tried to extend that a bit more by embedding p-tau into the picture. So amyloid, then p-tau, then tau. Where does ApoE4 modulate that? So does E4 lead to faster p-tau secretion or to a stronger aggregation potential of p-tau, so to say? So Anna, as I said, now postdoc in the group, has looked at that, and she's just plotted here amyloid against plasma p-tau here from ADNI and A4. In relation to E4, no difference. So the interaction terms are completely non-significant. So we don't see a different p-tau response in those people carrying that genetic risk factor. However, if you look into tau PET, there's a completely different picture. So here you look at how fast is tau accumulation in the presence of abnormal p-tau levels. Again, only look at this Q1 plot, that's the most relevant one. And E4 homozygotes show the strongest tau accumulation in the presence of abnormal p-tau. Heterozygotes or non-carriers show only fairly little accumulation. So there's a clear gene dose dependent aggregation potential increase of p-tau to fibrillar tau. So that means ApoE4 comes into play here at that particular part. So you develop p-tau, and then you're more likely to develop subsequently fibrillar tau pathology. Can we back translate that into clinical use? Because we're all often talking about thresholds and cutoffs, how do we define abnormality? But none of them takes into account individual factors. And we've, in fact, known that for a long time from regular or normal medicine, that we have different normality and abnormality cutoffs for, let's say, men and women or different age categories or whatever. So we looked here, do different p-tau cutoffs signal that abnormal tau PET increase depending on your E4 genotype? And you can already see here, this is the probability of transitioning to tauopathy in E4 homozygotes, heterozygotes, and non-carriers. Here ADNI, here A4. And having an ApoE4 allele shifts your p-tau cutoff to lower levels. So lower p-tau levels mean more in an E4 carrier than they do mean in a non-carrier. So we should really, as a field, think of, should we tailor biomarker cutoffs to at least these very common and very highly penetrant risk factors that we know? And maybe speculative, that is even one reason why from anti-amyloid trials, E4 carriers don't benefit that much, because at a given point of amyloid pathology, they might be further transitioned in terms of tauopathy and therefore experience less of an attenuation of clinical decline, because removing amyloid in those people, there's enough damage already that has happened. So, but that is an empirical question whether anti-amyloid treatment needs to come earlier, potentially at lower pathology levels in E4 carriers. So modulators, genetics, that's kind of that part of the story. And then one recent thing that came up more and more and more is co-pathology, because for a long time, our naive biomarker field, we knew we could measure amyloid, we could measure tau, and that's all we were interested in, not considering all the things we could not measure. And of course, the aging brain does accumulate various types of pathology, alpha-synuclein and TDP-43 being the most important ones. And so then CSF alpha-synuclein seeding aggregation assays came out, and it became quite clear in line with the postmortem work that these co-pathologies are highly prevalent. And here you just see people basically who are amyloid positive in orange, and the likelihood of their alpha-syn seed aggregation assay being positive. And you see in people with amyloid positive, so AD dementia, almost 50%. In MCI here, 20 something percent. Even in preclinical AD, already 20%. I mean, depending on the sample you look into, those numbers will vary. This here is just ADNI. But what is very important from a biological point of view is that alpha-synuclein has in cell and animal models shown to be a co-aggregation factor for tau pathology. So it's kind of a chaperone that helps tau misfold and aggregate quicker. So we were just wondering, do these people who have amyloid in their brain and who have positive alpha-synuclein pattern, do they aggregate tau faster? Long story short, yes, they do. Here, this is amyloid against looking at tau accumulation here for the temporal meter RI. Blue ones are alpha-synuclein negative. Orange ones are alpha-synuclein positive. And you can see that amyloid has a higher likelihood in triggering tau aggregation, more or less, if you have concomitant CSF or alpha-synuclein co-pathology. So that means also these co-pathologies, if we think of precision medicine in the future at some point, need to be factored in for determining the velocity of your pathophysiology overall. And this is just looking at their PET patterns, how they look like. So, for example, this is a beta positive, alpha-synuclein negative subgroup. Much less tau pathology than the positive counterparts. Very similar thing if you look at longitudinal tau accumulation rates. So again, the alpha-synuclein positive, amyloid positive, they accumulate faster. So that's the pathology side, but we can come back to brain network topology as the main routing factor for tau pathology throughout the brain. And this is an idea that we developed during COVID. So the brain's network architecture is not rigid. It's not everyone's talking to everyone. It's a very modular architecture where you have specialized networks, and everyone with experience in clinical neurology or neuropsychology should know that very well. Let's say we have a motor network, a visual network, or a language network. I'm just making things up, so I just sketched that outline. And the idea was then, so if tau pathology, if the epicenter emerges somewhere in the periphery, in a region that is not very well connected, so during COVID, let's say, tau was in home office, then it could only infect the pizza delivery guy. Yeah, so not very rapid spread. This is why we had curfews. Whereas if tau goes to a party, talks to everyone, then it should spread like wildfire. So basically, the idea is if tau emerges in a hub region, which is characterized by a lot of connections to the rest of the brain, that should lead to a faster overall disease trajectory. So in order to test that, we first of all looked at where in the brain are these globally connected hubs. This is here from a reference database of the Connectome project. And everything here in red orange is more of a hub region, and everything in blue is more of a non-hub, so more of that region. You can immediately tell the temporal lobe, typically affected by tau in old age AD, has very specialized connections, but not that brain-wide. And these more neocortical areas, which we typically see abnormal in early onset AD, they are hubs. So we then mapped the individual tau PET patterns to those hubs, and I'll skip the mathematics. It's not that complicated, actually. So we looked here, for example, this individual with a more temporal lobe tau PET pattern gets a tau hub gradient, we call that measure, that is more negative or lower. So tau is more shifted towards non-hubs. That individual with their tau epicenter being here on top in the parietal lobe gets a more positive tau hub gradient score. So it's just a measure of balance of your tau pathology between these regions and these regions, nothing more. And then if you look whether people who do have more tau in their hubs show faster tau aggregation, so global annual rate of change in the entire brain, that's exactly what you see. So people who have more tau in their hubs aggregate and accumulate faster, mostly in symptomatic AD. Here in the preclinicals, it was hardly there, and in Biofinder it was not there at all because those guys had no tau pathology really to begin with, so there's not much to model. But in asymptomatic people who have already substantial amounts of tau in their brain, or detectable, let's call it, there not only the mere presence of tau, but also where it is and how that epicenter is embedded in your overall brain network architecture tells you something about the velocity of subsequent aggregation and spread, and thereby on clinical decline. Is the type of hub important for how tau spreads? Because we know in the brain we have different types of hubs. That's more recent work from very connectomics nerdy guy in our group, Fabian, was a postdoc, and who thought that we have connector hubs in the brain that cross-link all the different modules. And if tau hits such a connector hub or emerges in close proximity, it should spread faster across modules, thereby fill the entire system more quickly. So this is just an illustration of that. So if you tau emerges closely to that connector hub, it should spread fast. Whereas if it hits a local hub that only connects within network nodes, then it should spread preferably within that network and have a more spatially circumscribed spreading pattern, and only move out of that network later on. So it's basically, he called it connector hub efficiency. How well does the epicenter connect to a connector hub versus a local hub? Does that, again, modulate how tau spreads? And this is where the connector hubs are found, typically in these association cortices or in the control networks that organize information flow across different brain networks, and the local hubs more within default mode network and so on, but that's not too important. But in essence, what he could show is that we're again always looking at that association between amyloid and tau rate of change. The more people have their epicenters connected to these connector rather than local hubs, the faster tau accumulation you see. Here, same again, validated in A4. The effect is not strong, not at all. I'm not claiming that it's a major driver of disease, but it just tells you that the detailed architecture of your brain can tell us something about how pathology spreads and unfolds. So with that, we've covered kind of the macro scale of how our brain networks composed, how do they look like. But now we can move back into the fine-grained element, which we think is really the major roadmap for tau pathology to spread, which is the synapse. And the synapse, bless you, I've mentioned that initially, is in healthy conditions versus abnormal conditions such as AD. At least early on, if you have amyloid, synapses are put into an hyperactive state, which may seem counterintuitive, but all the preclinical or the experimental evidence points towards that. There are different mechanisms suggested, one of them being whatever is cleaved off amyloid, which is a transmembrane protein. From that protein leaves a pore in the cellular membrane that is permeable for calcium. So there's a more constant calcium influx, therefore more action potentials. Amyloid can also directly bind to acetylcholine receptors, thereby triggering hyperactivity and so on. And very importantly, this is one of my favorite studies already from 10 years ago. This is a transgenetic tau model where they also modeled in optogenetics. And if they stimulated the hippocampus, that hippocampus accumulated more tau, meaning accumulation is activity dependent, but also the aggregation was faster in the contralateral connected hippocampus. So that hyperactivity may drive faster tau spread. And that was initially just an hypothesis. Now we try to more directly test that. We did unfortunately not have here any brain-wide markers such as EEG that could directly tell us about hyperconnectivity, hyperactivity. I'll come to that later. But I've talked to Kai Bleno actually when we were in China a few years ago, and he said, "Oh, Nico, look at GAP43. It's a protein involved in activity." And then I've looked a bit into it, and they've measured it at CSF. And this protein is highly expressed during development, and basically meaning your synapses are remodeling and they're in a hyperactive state potentially, and it's upregulated in epileptic conditions. Meaning concentrations of that protein could tell us potentially that synapses are remodeling and they're remodeling in a hyperactive way. And what speaks for that is that this marker, which Kai's lab measured in AD patients here, is really strongly upregulated in Alzheimer's disease patients compared to non-AD conditions such as Parkinson's disease, synucleinopathy, no upregulation at all, nor in PSP, a primary 4-repeat tauopathy, but you find elevations in dementia with Lewy bodies. Why? Potentially 50% of those cases have concomitant amyloid pathology, and corticobasal syndrome is either a primary tauopathy or atypical Alzheimer's disease. So it does make sense that you find an upregulation in exactly those people here. Meaning, piece of evidence number one, amyloid potentially makes your neurons somewhat hyperactive, and that could be reflected in GAP43. We again looked into our individualized spreading models, Looked here again, spread from the tau epicenter to here, this Q1 region, depending on whether you had, it's maybe a bit hard to read, high GAP43 levels in your CSF, everyone in orange, or low GAP43 levels in blue. And you can see again that tau only really starts accumulating strongly in those people in which this marker that potentially has to do something potentially with synaptic hyperactivity is increased. And that this effect goes away the further you go away from the epicenter, so to that region, meaning potentially that has to do something with transneuronal tau spread. But we didn't want to stop here, but think of it one step further. So can we actually look at this hyperactivity somehow? Where the idea is amyloid develops here, let's say in your medial parietal and medial frontal lobe, tau somewhere at a different place in the temporal lobe. But of course, neurons are not locally restricted to a particular region, but they have axons. That's what people often forget about, that there is a connection between them, or the same neuron has its soma here and its dendrites or axonal end feed over here. And that amyloid may lead to an overdrive of that connection, opening the gates and then leading tau to spread out to those regions. And then ultimately, when degeneration kicks in, that connection breaks down. So we tried to test this. Does amyloid induce hyperconnectivity? Because any hyperactivity occurring over a longer time should trigger long-term potentiation, therefore coherent co-fluctuation of neuronal activity leading to a connectivity increase. So Sebastian, a clinician scientist in our group, looked at this and basically he thought, does amyloid trigger, if I look at both ends of the connection, look at amyloid at both ends, does that lead to an increase of that connection? And does that increase of the connection lead to a faster travel of tau from the beginning to the end of the connection, so to say? So these are the regions when they were connected to the tau epicenter where he found a connectivity increase. So quite a broad effect of having higher amyloid, having higher connectivity. Again, that may seem counterintuitive and may explain also the vast heterogeneity in the fMRI findings on Alzheimer's disease. One saying upregulated and the other say downregulated, blah, blah. You need to look at it closely in relation to pathology and, of course, have a hypothesis and not just look at it exploratory. And basically what we found is amyloid leads to an upregulation of a connection. And that upregulation of the connection, then this is really this mediation effect, that upregulation leads to faster tau accumulation in the downstream region. So basically the whole pathophysiological model put together in one framework. And since hyperconnectivity is again not necessarily hyperactivity or hyperexcitation, so that remained to be tested too, we've started looking into a more complicated model, a neuronal model or model of neuronal activity developed by or applied here by the group of Thomas Yeo. It was developed by Gustavo Deco, where you can use fMRI data to get an idea of the excitatory to inhibitory balance. And this is work in progress, but what again Sebastian finds is that if you look at a range from low to high amyloid levels, the excitation to inhibition ratio goes up. Very later, it goes down again. Yeah, but this hyperexcitability here in the beginning may be really a gate opener for a tau to be secreted and spreading. So with that, come on. Now, that's the potentially intermediate summary slide, or so I can end here either, or tell a bit 5, 6, 7 minutes more about primary tauopathies. What's your opinion? Okay, more about primary tauopathies. It's very similar. Yeah. 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 Well, functional MRI data, of course. Yeah, we look at activity, so we rarely, I'm too stupid for diffusion MRI, so I don't tend to use it that often. So take home from all the rubbish I said is tau spreads from epicenters across connected regions. It's driven by amyloid and p-tau increases, and that the underlying mechanism might be abnormal synaptic activity, hyperconnectivity, and in the end, hyperexcitability, all circling around that mechanism. That the rates and patterns of tau are not the same for everyone, but they're influenced by your brain network topology and where in that brain network topology your pathology emerges. And that co-pathology is genetic risk. All of them influence our disease trajectory. So these Jack models that we have are, of course, they need to be an oversimplification. And within that, there's a lot of heterogeneity that we can try to systematically and mechanistically explain. And I think that has a few opportunities, namely if we know the modulators of, in the end, what's the amyloid cascade, we can try identify treatment targets that translate into a breaking of that amyloid tau cascade beyond just removing amyloid, because that's certainly not the best thing to do in every patient. And also knowing in an individual when and how tau spreads can help us decide when to treat, let's say, with an anti-amyloid drug or whatever is going to come next. But tau is not exclusive to AD. It's the isoform kind of is, the majority of being 3 to 4 are tau, but tau is also the major driver of primary tauopathies. And here I come to progressive supranuclear palsy and corticobasal degeneration, PSP and CBD, which are 4 repeat tauopathies. So their isoform looks different, and they lead to mostly what we know as Richardson syndrome, so postural instability, falls, vertical gaze palsy, or corticobasal syndrome, which, caveat, can be Alzheimer's disease. So if you want to study it in a 4 repeat tauopathy, make sure that these patients are amyloid negative. And what is very interesting about these patients, or these diseases in general, they have a completely inverted tau pathology spreading pattern compared to AD. AD is a mostly neocortical or cortical tau pathology, whereas PSP and CBD are mostly subcortical. So they start in the brainstem, then the basal ganglia are heavily affected, and then in the end it moves to the cortex. And this is also why they associate with atypical Parkinsonian syndromes rather than more cortical symptomatology. What the problem was, or still kind of is that measuring 4 repeat tauopathy, the gold standard is, of course, post-mortem, and tau affects neurons and glia, mostly neurons, but glia as well. And the tau tracers didn't see this tau pathology, potentially mostly because fluorotaucipir has massive off-target binding in the basal ganglia, which are exactly the target site of that 4 repeat tauopathy. So luckily, my colleague Matthias Brendel from Munich got into great contact with live molecular imaging, now Lantheus, and we started now 7, 8 years ago, or even longer maybe, scanning with their PI2620 tau PET agent. And we found signal increases, which were exactly where we expected them to be in PSP, Richardson syndrome, non-Richardson syndrome. In the controls, you don't find that. And also in here, Parkinson's cohort, you don't find that. So we thought, okay, this might be specific. Autoradiography studies suggested, yes, also co-localization between where there's 4R tau and where there's tracer binding. So we could do exactly what we did in Alzheimer's disease, now using their tau PET imaging data. So this is just a mapping of here a cohort of patients with a clinically suspected primary tauopathy. So where we had a diagnosis of corticobasal syndrome or PSP clinically, and they were amyloid PET negative. So the underlying cause is likely to be 4 repeat tau. It's not 100% sure, but it's likely. And this is just the mapping of where their tau epicenters are. And you can see, aha, here, they're in the subcortex mostly. And then later on, some of it in the cortex as well, very well matching their clinical phenotype. And then we did exactly the same as we did in AD patients. Regions that are more strongly connected to each other show more similar uptake in tau PET. Yeah, so that is basically telling us, again, the brain network, because we think tau spreads also in these individuals across synapses. So we would have expected exactly that pattern, and that's what we found. But caveat, which I mentioned initially in those patients, is those diseases affect neurons. So we find tau in them, astroglia, and oligodendroglia. So where does your signal come from? Because looking into a voxel is, of course, a mix of all of that, plus unspecific binding. And that tracer is certainly not ideal for those diseases. So we got in touch with Gabor Kovac from University of Toronto, who had loads of post-mortem data from patients like that. And so we tried to replicate everything that we did with PET, now with post-mortem data. For that, we needed to have regional sampling, so a bunch of different brain regions from which we had ratings of no, low, medium, high for repeat tau with a clinical, so a post-mortem secured diagnosis here of PSP, and test whether we find, again, between brain regions, does the connection matter about the covariance and tau accumulation, and is that mostly driven by tau that you find in neurons, because they make the synaptic connections. So back then I sat down together with a neuropathologist and we were really hand drawing where they take their probes on an MRI scan, because we needed to map the post-mortem protocol back to our brains, because from dead people you can't get connectivity. So we need to have that from healthy individuals. And we had roughly 100 cases from Munich, roughly 100 cases from UPenn. And then we did exactly the same as for PET. And these are the two different samples. And maybe you can't read the X's, so I'll read them out for you. It's connectivity versus covariance in post-mortem tau load. Connectivity in healthy people, covariance in tau or similarity in tau from these 100 dead brains. You see again, those brain regions who connect when you're healthy show similar tau levels when you die of PSP. Very similar, different spatial pattern here for the regions of interest for the UPenn dataset, so very nice cross-validation. As a reminder, this is the PET stuff. Very similar associations in vivo post-mortem. And then we repeated everything, and this is really too small to read, for astrocytes, oligodendroglia, and neurons. And you see a stepwise increase for neuronal tau. So that seems to be here the strongest driver. Here it's a bit less of a difference. Oligodendroglia follow. And that could make sense because the oligodendroglia follow the axonal outline, basically. So their tau may just, and there has been evidence that tau can move out of the axon and be taken up by the surrounding oligodendroglial cell. And here I think I'll end and skip that perfusion part and yes, show basically the same conclusion slide as for AD, just that brain networks generalize as roadmaps for tau spread across secondary tauopathies, which is AD, and primary tauopathies, that we have in vivo and post-mortem evidence for that, and that the brain's network is an important vehicle for how your disease spreads. And this is just a summary of how we try to approach our entire research. So we're really trying to understand mechanistic disease models and translate them into patient-centered prediction and incorporate modulating factors and then basically translate it to primary tauopathies. With that, I would like to, no, this is important, thank everyone here in my group who do all of that, because I can only quote Bill Jagers, who says, I just answer emails. It's kind of true. And all our collaborators from across the world, and hope I didn't bore you too much, and thank you.
Thank you, Nico, for the wonderful presentation. I'm sure that a lot of people in the room and also in the virtual room will have a bunch of questions for you.
Don't roast me.
So
break the ice. I'll break the ice.
Yeah.
So I have one more methodological question reflection, and one more biological mechanistic. So the methodological, since you, so first of all, great talk, great picture of all your work. So I really enjoyed the talk, and thank you for the delivery also of all your data. So the methodological consideration is, so you have use a lot of functional connectivity measure, and you say, okay, this biological aggregation depends on functional connectivity, and on the other side, it also depends on activity.
Yeah.
So, and in neuroimaging, of course, there is always the problem that the measures and the biological meaning, they are ambiguous, right?
Yeah,
Functional
exactly.
connectivity is a measure that not necessarily reflect an actual biological connectivity.
That's true.
It is confounded by activity.
Yeah.
Then you enter rapidly into a circular situation where there's how can you claim, and in some of the analysis there is probably not a problem.
Yeah.
Sometimes they may.
Yeah.
If there is a change in local activity, it will change the measure of functional connectivity without any actual change in biological connectivity.
That is a very good point, and I know where you're going, and interrupt me if I'm not giving the exact right answer. So the circular logic is really a problem. This is why for most of the analysis, where we just look at the brain network as like a grid for pathology spread, we always use the brain network template from healthy individuals for which we know there's no tau and there's no amyloid pathology. So there should not be a confound. Only in very few studies where we look at more of connectivity changes as an endpoint. There we use individualized fMRI data, knowing about the caveats, of course. So that very last, or one of the last STM studies where we looked at hyperconnectivity, there we specifically look into how does amyloid promote connectivity changes, and if we observe them, do they themselves promote tau accumulation and spread? This is one of the few studies where we've used individualized resting state fMRI. We're sometimes asked by reviewers, can't we repeat that with individual fMRI? Sometimes we do in a supplementary, but bringing that exact argument that you brought up, I think, so the pathology spreads when the brain network is ideally still healthy, and then you should see downstream impacts of the pathology who make you enter that circular logic. So we're trying to avoid that by moving away from individual data. Often.
Okay, so, and I mentioned this because I've been working a lot on this methodological approach to disambiguate connectivity
and activity
from actually local activity.
Yeah.
We have a nice paper on the problem.
Yeah.
So if you're interested, I can.
Yeah, absolutely. And maybe also commenting on that because that's what I forgot. Connectivity, we used functional connectivity in the very beginning because the animal data showed tau spread is activity dependent. So functional connectivity seemed to be the more logic measure to look into rather than just, is there a diffusion-based connection between A and B? Next point you mentioned, there doesn't need to be necessarily a direct underlying connection. Of course not. That every connection we look into is highly likely to be multi-synaptic, but I don't think that's a big problem. But coming back to the initial problem that you phrased, we're currently connecting, this is my in-house cohort, where we're collecting 30 minutes of resting state fMRI in each individual to really look into that combined with EEG, how do connectivity and activity dissociate or associate depending on your biological status, because I think that's unaddressed still.
And I transition the more biological question, which is about a reflection on the concept of spreading.
Yeah.
Because, of course, the great majority of the data are available, even in humans or in animals. Yeah. Spreading is defined at very, very, very slow time scales.
Yeah.
And you make a strong assumption, explicit, that the spreading at the fast time scale goes in the same direction of the spreading of the slow time scales. I make an analogy to make my point. So traffic jam.
Yeah.
The spreading of the traffic jam goes in the opposite direction if you see it on a slow time scale of the spreading at the direction of the cars.
Yeah.
Right? You have a block and the jams spreads in this direction.
Yeah.
So what is, and on the other end, I did a lot of experiment with alpha-synuclein in mice.
Yeah.
So in the Parkinson disease field, there are few contradicting or puzzling findings. So for example, if you fill with viral vector neurons of alpha-synuclein, mutate and non-mutate, and it does not cross to the next step, which is, if it's spreading, why is not going there? And on the other hand, if you start putting fibrils or stuff either above or in the periphery, they go in both directions.
Yeah.
So in reality, the real direction of the spreading in the fast scale could be either way, independently.
That could be either way, yes. And we're not making any assumptions about the directionality of the connection itself, because fMRI per se is undirected. I mean, there are these directional measures, but not a big fan, and not, or let's say, not convinced by their biological plausibility.
Because when you
it's a
in the postsynaptic neuron, you're clearly saying
that's true. That's true. And that's most likely. I don't know whether it's probably not like always in biology. There's a prior probability distribution, but both ways are potentially possible. Whichever, I would assume that from the experimental models that we have, and also from looking into, because we see tau PET locally emerging in one focal region, and then it goes basically to a connected partner. If it, I'm not sure whether I'm answering that right, but I totally get your point, and the directionality of the connection we can't infer from fMRI data. Long story short,
Thanks so much.
You're welcome. If you have an idea, however, then I'm happy to talk about it.
Yeah. Thank you again for your talk. Now, many years ago, more than 10 years ago, some people from UCL suggested a concept that I thought was quite accurate, of molecular nexopathies.
Yeah.
I mean, that term. That was not a successful term because nobody talks about that. But the concept was very,
I know that.
Was the beginning of this way of looking at the diseases. But in this concept, they considered that these diseases progress along anatomically connected regions and towards vulnerable connected regions. That means that not all connected regions develop disease, but that there are vulnerable or more vulnerable and less vulnerable regions. We see that, we see that in particularly in diseases where you have different strains, like in prion diseases or now even in tauopathies. Yeah. So I was just asking myself the way you could search for vulnerability factors within your models.
Yeah, exactly. It's a very, very good point. We've actually, with Jake Fogel, we've written a review a few years ago, which is Nature Reviews Neurology, also addressing that point to a certain degree. So I think amyloid is a vulnerability factor, let's say. And so you have tau, let's say, somewhere in the temporal lobe, and then you develop amyloid elsewhere, and that increases your vulnerability, if you will, for tau to spread from A to B, regardless of the mechanisms behind it. Second thing that has been shown quite some time is that gene expression matters. So if you have another connected region that does express, let's say, MAPT to a higher degree, and then there is more biological substrate to transform into something that is abnormal, I think that plays a role. And yes, there is clearly vulnerability patterns. And I think if you have a spatial map or an idea of where something vulnerable is or not, then you can always test whether your vulnerability increases your prediction accuracy, kind of correlating or correcting for a spatial autocorrelation. That is always some sort of an issue in fMRI or MRI data in general. But yes, then that's clearly something to look into. And I think there are a lot of vulnerability factors to incorporate. And amyloid is, you can phrase it as a vulnerability factor. Not everyone would like that term though, but yeah.
Okay, thank you.
We will now read one question from the platform, which is very interesting. I also thought about the same. And it's Mark Ryu. He asked, he says, thank you so much for the talk. You mentioned that A beta could be behind the increase of activity, which in turn will also increase tau spread with activity. What about the spread in primary tauopathies? Could pathological tau also increase the activity by itself?
Yeah, could be. And there's just been a paper out from Susanne Wegmann and another one from Mark Busche from earlier last year showing that tau does have an impact on neuronal activity itself. but very complicated and potentially in both directions, both increase and decrease. I have no clue about how that would play out in primary tauopathies, and I think it's very complicated. We don't have good subject level data yet to look into that. Second point is brainstem, and if you would need to look at the very early stuff or pathology, then you would need to look kind of brainstem, subcortical, where fMRI isn't the easiest. Very interesting, but I must say, no idea, but could be. Yeah, it's a valid idea.
As a suggestion, the data that I presented in HAI about the amyloid negative, tau positive cases in ADNI, this may have also fMRI.
Yeah, that's true. There are, however, few, and ADNI, in our paper, we've carefully selected people with a good fMRI protocol because it's multi-centered, so you have to be careful. I mean, we're recruiting these patients at home in Munich, but I think we would need somewhat like 50, 60 people to be robustly powered to look into this somehow. And once we have that data, we will certainly do that. Michael is texting me in English, which is weird.
Brought in the. Also, I had a question. I already thought about this when I read your first papers, and I found a bit of a paradox related with education.
Yeah.
So we know or assume education is a protective factor in AD. We also know it increases functional connectivity.
Yeah. Oh, yeah. This is what I did my whole.
I wonder how this dissociates, how you include this in your analysis, and if you have studied education isolated from.
In fact, we did. I did my entire PhD on cognitive reserve and resilience, and looking into brain mechanisms, how education would guard you against developing dementia, or AD, the clinical syndrome at least. So education is more complicated than increasing your connection in general. It's more of, in graph theoretical terms, of your overall network topology, which may be a bit more efficient. There are different ways, all of which are non-ideal, which try to break that down in one number. And one of them being brain network segregation. So to which degree brain networks segregate into functional modules, which is considered a more efficient brain network topology. If that is entirely true, I'm not claiming that, but let's say that more efficient brain network organization would protect you against dementia or developing AD dementia if you have the pathology. So we looked at this in CIEN. That was in 2016 or 17, and we found that people with higher education levels show more brain network segregation. That means potentially they have a higher connection here and there, but overall their modules are functionally more isolated from each other and have only specialized pathways cross-linking them. And we found that the more segregated their brain networks were, the less fast they declined cognitively. And then back then we had no clue about tau PET or anything yet. So we thought, okay, they have more efficient brain networks, and therefore they decline faster. They can maintain cognition longer because their brain networks operate more efficiently. But then a few years later, we actually thought, okay, hang on. If these modules constrain tau spread, and if a module is more isolated with only a few very efficient connections moderating between these modules and exchanging information, then it's like this connector hub thing that I showed. Then the likelihood of tau to spread from A to B should be lower. So we looked at, does this brain network segregation actually attenuate the spread of tau? And it does. Or it's associated with that. I'm not making causal claims here. So that could be one cross-link where how education could not only, I think education is at play two ways. First of all, neuropsych is always a bit biased. People who are more intelligent or educated work better on those tasks. Yes, education adjusted norms, but still. That is one thing. So it may buffer your effect of pathology and cognition, but it may also buffer the unfolding of pathology itself. And I think there's been one or two papers recently showing that there is an effect of education on amyloid-related tau aggregation. That one was from Tengfei Guo's group in Shenzhen in JAMA Neurology, I think. I wasn't too convinced by the spatial patterns, but
I think we tend to oversimplify a lot the education.
Yeah, if education attenuates your development of disease, you must try to find an explanation kind of for it, rather than 70-year-old patient, go to school. It doesn't work.
Also, I also wonder when you work with amyloid PET, maybe going to the beginning of the talk when you showed the mediation of p-tau between amyloid and tau, which is very interesting. You tend to work a lot with centiloids, and I understand why is that, but also amyloid PET provide us with a very limited but still some extent of spatial information. And I wonder because if you had studied about the co-localization of amyloid in certain regions and later tau spreading in
Yes. This is work in progress again by Anna Stewart, and also so two main findings here. Co-localization is important because I think ultimately you can enter the AD trajectory in two ways. You can either have first amyloid at a fairly young age, then amyloid has the potential to generate its own tau, and then it's very likely to generate that was my assumption, where there is amyloid, because amyloid can lead to tau hyperphosphorylation. And this is exactly what we see in early onset AD patients who develop amyloid at a younger age, enroll or enter that, or DIAN patients, let's say. They have a much stronger co-localization between amyloid and tau than people with very late onset AD. And here I think that should end this which comes first, blah, blah debate. I think you can have fibrillar tau in your temporal lobe, wherever, before you have amyloid. And that sits there. And then amyloid comes in and amplifies what is there. And just, again, thinking of spatial priors, you may have just a higher likelihood of having tau as you age in your temporal lobe than amyloid comes in neocortically. And this is why the older people are, you see more of that typical temporal lobe pattern. The younger people are, I'm talking about symptomatic people, the more neocortical and upward shifted that pattern is, co-localizing with amyloid. Also, that co-localization is increased in E4 carriers, which speaks into that very same direction. Yeah.
Thank you for the presentation. It made me think about the myelin.
Yeah.
Because the patterns that you show were very similar if you compare them with the myelination patterns. And it was really interesting because I read that the regions with a similar myelination also have a similar similar levels of functional connectivity. And it was really interesting because there is a parallelism with the things that you were explaining. And also with the patterns, because the regions with less myelination are more vulnerable to the tau pathology.
Yeah, that's true. This is on, again, you're picking up something that Bragg noted already in 91 in his paper, that there is an inverse, or Bragg stages on average look inverse to the myelination pattern during brain development. That's completely true. And in fact, we've done, or that was my former PhD and postdoc supervisor, Michael Evers, he's done those studies, seeing on average, if you look at a myelin template, yes, you find exactly that. Myelin quantification in vivo is, however, very difficult and very tricky. There's no good measure. T1 over T2 contrast or the other way around, that's not great. Then there is these McDespot sequences, and then there's a couple of other things. We've tried in Munich, or Michael has tried in Munich now for 2.5 years to implement myelin sequences. Now they may come and tell us more about that, because yes, in the end, that might be some important modulator. And just really speculating now, let's say you have high myelination protects you, or is protective against tau at either end of the connection, whatever. Let's say you have a very highly myelinated neuron. That neuron is, of course, more efficient in transferring information, so it needs less pressure on the tube to have a signal coming out of the other one. So maybe that is an indirect mechanism constraining or making a synaptic transmission more efficient, therefore leading to less tau spread from A to B. Highly speculative, but could be. But in our in-house study that I mentioned, we're trying to now-ish start with this, I don't know what it's called, McDespot, whatever sequence from some guy from the NIH, which takes 20 minutes to acquire. It's very complicated and makes a lot of assumptions. They've done that using, with QSM, you can also do chi separation, and they've done that now in Biofinder, and the results are very mixed. But again, myelin is probably very difficult to measure, and probably someone being more familiar with MR physics, maybe you can tell us more about that. Okay.
Thank you.
Yeah, you're welcome.
You have beautifully presented that the spread of tau is related from extent to connectivity.
Yeah.
Question is, could you estimate, according to your data, the correlational studies, to what extent this factor, this connectivity is the main, the almost universal factor determining the spread, or should we also consider other?
Yeah, I think other things as well. So we're again, we're alluding to lots of work that we're doing at the moment. I think spreading, and with PET imaging, we're looking back in time a lot. Spreading is something that is happening very early. It's like when your colleague was talking about temporal scales. We cannot identify that very rapid temporal scale. I think spreading is happening very early, and then tau enters an auto-replication cycle at some point, once a critical mass is reached, and we've tried to quantify when that happens now. So tau, or connectivity, always determines the pattern, but it's not necessarily that connectivity is always causal at every point in time. It may determine the pattern very early on, and then a critical point is reached and tau just starts auto-aggregating, and the pattern that you see is just like the leftover from what connectivity, the distribution that it has determined in the very, very, very beginning. It's maybe a bit complicated, but I would in essence say our work from atypical AD shows that the patterns are overall universally predict or define, constrained by the connectome architecture. Other things contribute as well: gene expression, vulnerability factors that have been mentioned, individual amyloid patterns that you have mentioned. All of that would lead to an R squared of 1 potentially at some point, explain all the variants. I think connectivity is very essential, but it does not mean that this is a suitable, let's call it, treatment target at every point in time. And we've quantified it, trying to pinpoint the amyloid PET threshold until which tau spread, or the accumulation itself, is amyloid dependent. Yeah, only once you've reached the tau positivity threshold, then afterwards it accumulates on its own, more or less. And that converges very well with the donanemab data saying, well, if you're full of tau, removing amyloid is kind of pointless. That's maybe a part of the answer.
I think that the connectivity determines the map.
Yeah, exactly.
And there are other things that determine other important
exactly
things, like the intensity, the
exactly. Think of
the effects.
Think of the very, very early days of COVID. There, the flight map from, where was it? Chinese city, I forgot the name. The flight map to Wuhan, thank you, to the rest of the world determined the initial map, but then local factors took over. And because there was only maybe 1 or 2 patients coming in, not just 100 a day from Wuhan. So the initial, and then at some point things dissolve and other dynamics take over.
And this is, in your opinion, congruent, concordant with the proposal of the black states in the
In a way, what Braque did, if you look at his papers, he averaged across a fairly high number of individuals. And if you take photos from everyone in the room and generate an average face, then there are 2 eyes, a nose, and a mouth, and 2 ears. But hidden in that group average pattern is a lot of heterogeneity, which a mean value will never show you. And he has, as far as I know, not shown a standard deviation map of how these patterns look inter-individually. So overall, the Braque patterns, if you look at group mean data, you'll find them. If you look at individual data, you see substantial deviations from it. Yeah, exactly.
There is, okay, there is.
Hi, Nico. Thank you so much
Hello.
for the presentation. There was a lot of mention on different vulnerability factors that influence the spread of tau, and I was wondering what role does vascular health play in this picture, especially white matter hyperintensities? How does that affect tau spread, and how does that really propagate?
Yeah, very good point, and we've tried to get a good idea of that for a long time. Overall, I don't see a good effect or moderating effect of your white matter hyperintensity load on how fast your tau spreads. There might be 2 things. Either your vascular health affecting your overall axonal system leads to de-differentiation, and that could lead to faster spread, or you have, let's say, a cut in a connection. That cut in the connection interferes with tau spread. So thinking of surgery to isolate, let's say, your entorhinal cortex, if you're positive, that would be kind of the experiment to do, which is unethical, but still, to see whether that interferes with tau spread. Vasculars, we don't see any good indicator that vascular comorbidities, at least to the degree to which we can measure them, have a strong effect. I think there might be some, but studies are very heterogeneous. Some find it, others don't. We've looked into 3, 4, 5 different data sets, never seen it consistently. So I've seen in one sample in the one direction, another sample completely the opposite, and then I don't know what to believe. But it's an unsolved question, I believe.
I wonder, there is a very speculative comment, but there is a subset of populations outside that don't have a medial temporal lobe that are patients.
I know. Yeah.
Surgery for refractory epilepsy. I wonder if you know about any paper that has looked into.
Believe me.
For example, looking for the 2 hemispheres if the tau spread. You don't have an.
Believe me, I've been talking, this is 5 years ago, to our neurosurgery, whether we can identify people who have been undergoing that type of surgery 20, 30 years ago, because that's typically a surgery that you don't do in a 70-year-old rather than young people. And then you have to find them 30, 40 years later, factor in the probability of having developed amyloid and tau. So very difficult to find, but super interesting. I don't know, is there anything out on that? That would be the idea. Or callosotomy, which has been done before.
No, I was actually asking.
No, okay, no. No, our neurosurgeons know about it, but no one's doing anything. Probably if we looked through the entire database, we'd probably find an N of 1. Yeah. Do you want your temporal lobe to be cut out and then we wait for 50 years? I'm dead by then, most likely.
Any more comments or questions? Okay, so we are well over time, so
yeah,
we may
sorry.
Thank Nico again.
Is there? Okay. Yeah, that was the only one online. Good. Thank you.